I was just reading the latest clinical data on central insulin resistance, and it changes how we should evaluate cognitive fog in healthy aging adult populations. Many healthy adults experience a persistent feeling of mental cloudiness and reduced sharpness, assuming it is merely a natural consequence of aging or poor sleep as we navigate healthy aging during daily cognitive tasks. We commonly attribute this fatigue to high stress or general metabolic sluggishness, without considering the specific signaling pathways operating within our neural tissue in our daily lives to protect our brain structure. However, neuroscience suggests that cognitive fog may relate to how effectively our brain cells respond to localized insulin receptor signaling pathways in the brain within neural tissues as we navigate healthy aging. Imagine a system where your brain cells struggle to receive critical metabolic signals, impacting their ability to process information and maintain synaptic plasticity under these metabolic conditions in our daily lives. We are exploring how central insulin signaling supports cognitive performance, helping to reframe mental fog from a vague symptom to a specific metabolic pathway change to support cognitive endurance within neural tissues. When insulin receptor sensitivity in the brain declines, neurons struggle to coordinate the rapid electrical changes required for memory and learning tasks as we age over time under these metabolic conditions. If we treat this fog only with caffeine or sleep, we ignore the local metabolic pathways that govern synaptic communication and cognitive endurance in healthy adult populations to support cognitive endurance. The stakes are high because central insulin resistance is associated with reduced synaptic density and accelerated cognitive aging over the long term of life for long-term health span as we age over time. Understanding the physical pathways of central insulin signaling allows us to investigate practical, low-risk protocols that support our brain's metabolic health naturally during daily cognitive tasks in healthy adult populations. The problem is that we often conflate central insulin signaling with peripheral blood sugar levels, ignoring how the brain regulates its own receptor sensitivity to protect our brain structure for long-term health span. By identifying the specific cellular mechanisms involved in neuronal insulin signaling, we can establish better daily protocols to support cognitive longevity and focus as we navigate healthy aging during daily cognitive tasks. To understand this metabolic process, we must define neuronal insulin signaling, which is the activation of insulin receptors widely expressed throughout the brain in our daily lives to protect our brain structure. These central insulin receptors are highly concentrated in regions like the hippocampus, which coordinates memory consolidation and spatial learning in healthy adults within neural tissues as we navigate healthy aging. Unlike in the rest of the body, insulin signaling in the brain does not primarily regulate glucose uptake by the active neural cells under these metabolic conditions in our daily lives. Instead, central insulin signaling acts as a neuroregulatory pathway supporting synaptic plasticity, which is the ability of connections to strengthen over time to support cognitive endurance. This plasticity is essential for encoding new information and maintaining fluid cognitive performance as we navigate various tasks throughout our daily life as we age over time. Insulin reaches the brain by crossing the blood-brain barrier via a specialized transport system that regulates its concentration in neural tissue in healthy adult populations. Once inside, insulin binds to receptors on neurons and glial cells, initiating a cascade of chemical signals that support cell survival and connection stability for long-term health span. During healthy aging, changes in receptor structure or transport efficiency can gradually reduce the brain's sensitivity to these vital insulin signals during daily cognitive tasks. As we age, persistent metabolic stress can lead to a gradual downregulation of insulin receptors on the surface of our neurons and astrocytes to protect our brain structure. This downregulation reduces the cell's response to circulating insulin, a state known in clinical literature as central brain insulin resistance, as we navigate healthy aging. When central insulin signaling is reduced, the pathways that support long-term potentiation, the cellular basis of memory, begin to operate less efficiently in our daily lives. Neurons struggle to insert new receptors into the synaptic membrane, which is a key structural step required to strengthen neural connections within neural tissues. Without this structural reinforcement, the connections between neurons remain weak, leading directly to the felt experience of mental fog and slower recall under these metabolic conditions. Furthermore, oligodendrocytes require insulin signaling to synthesize the lipids needed to maintain the protective myelin sheath around signal-carrying axons to support cognitive endurance. A reduction in insulin sensitivity can therefore affect myelin maintenance, leading to slower conduction speeds along white matter tracks in the prefrontal cortex as we age over time. This dual impact on synapses and myelin structure explains why central insulin resistance is a key contributor to declining cognitive endurance in healthy adult populations. Microglia, the resident immune cells, are also affected by insulin signaling, which normally helps maintain their resting, non-inflammatory state in the tissue for long-term health span. When insulin signaling is impaired, microglia can become primed, releasing inflammatory cytokines that further disrupt synaptic function and local cellular performance during daily cognitive tasks. This local inflammatory feedback loop can sustain a state of metabolic strain within the neurovascular unit, limiting nutrient delivery to active neurons to protect our brain structure. The brain must expend more resources to perform routine cognitive tasks when these local metabolic signaling pathways are operating below optimal levels as we navigate healthy aging. Basal metabolic efficiency is closely tied to how well our brain cells respond to these localized receptor inputs over the lifespan in our daily lives. Ensuring robust insulin sensitivity in the brain is therefore essential to protect the physical infrastructure that carries every signal we process within neural tissues. We must examine the boundaries and limitations of this metabolic model to understand its practical application in healthy aging protocols under these metabolic conditions. One limitation of central insulin research is that we cannot directly measure real-time receptor sensitivity in the brains of living human subjects to support cognitive endurance. We rely on indirect measures such as intranasal insulin challenges, systemic metabolic markers, and cognitive performance tests in clinical cohorts as we age over time. In some clinical studies, improvements in systemic metabolic health are associated with better cognitive scores, though individual baseline sensitivity varies significantly in healthy adult populations. Additionally, we must recognize that brain insulin resistance is not the sole cause of fog, as sleep debt and stress also contribute for long-term health spans. The population boundaries of current human studies are narrow, and more research is needed to isolate central from peripheral metabolic changes during daily cognitive tasks. Because we cannot directly observe microscopic synaptic changes in humans, we must interpret imaging and biomarker data with appropriate scientific caution to protect our brain structure. Therefore, we analyze central insulin resistance as a significant contributing association in cognitive aging, rather than an inevitable cause of decline as we navigate healthy aging. Answering our opening question, cognitive fog is often a localized metabolic signaling issue rather than a sign of inevitable structural decay in our daily lives. By understanding this physical mechanism, we can reframe how we support our brain, focusing on systemic metabolic stability to protect neural connections within neural tissues. When central insulin sensitivity is supported, synaptic plasticity remains high, helping to maintain executive function and cognitive endurance during daily work under these metabolic conditions. The practical challenge is supporting brain metabolism through non-invasive daily protocols dietary habits, and physical activity as we age to support cognitive endurance. While we cannot reverse all age-related metabolic changes completely, we can optimize the systemic factors that influence central insulin receptor sensitivity as we age over time. Consistent physical exercise, for example, is associated with improved systemic insulin sensitivity, which supports the transport of insulin across the barrier in healthy adult populations. Avoiding frequent glucose spikes helps maintain stable receptor expression, reducing the risk of downregulation in both peripheral and central tissues for long-term health span. These metabolic habits do not target memory storage directly, but they support the cellular environment required for efficient synaptic transmission every day during daily cognitive tasks. Supporting central insulin sensitivity requires a unified focus on dietary consistency, regular movement, and proper sleep to optimize these vital pathways to protect our brain structure. To apply these concepts, consider monitoring your cognitive endurance during sustained tasks. Notice when fatigue begins as we navigate healthy aging. Ensure consistent physical movement is integrated into your daily routine to support systemic metabolic health and receptor sensitivity in our daily lives. Focus on consuming nutrient-dense meals that promote stable blood sugar levels, helping to avoid rapid fluctuations that impact cellular performance within neural tissues. Prioritize quality sleep to facilitate the natural metabolic clearance processes that occur in the brain during deep, slow-wave sleep cycles under these metabolic conditions. Consult a qualified healthcare professional to monitor metabolic biomarkers like fasting insulin and HbA1c, as systemic health supports brain function to support cognitive endurance. Avoid self-prescribing restrictive diets or metabolic supplements without professional guidance, as individual physiological requirements vary significantly among adults as we age over time. Focus on establishing simple, low-risk daily habits that support metabolic efficiency and reduce systemic cellular stress naturally and safely in healthy adult populations. Track your daily activity, dietary patterns, and cognitive focus to build a personalized, evidence-bounded framework that supports your long-term health span for long-term health span. Optimize the protocol, protect your brain, and I'll see you in the next data set.